We will now have our question and answer session. I invite all the speakers up to the stage, and if you do have a question, please introduce yourself. Oh, here we have a question. Cool. Go ahead.
Thank you so much, Anna here. I have a question for Alessandro. Can you give an example of sham meditation? What do we think?
Yes, so a sham meditation is a type of meditation in which participants is induced to think that is actually doing meditation or mindfulness, while there are no active components of mindfulness. So no focus on the present moment, no acceptance of inner thoughts. So in this type of meditation, usually the relaxation part is kept the same, but you just remove all the active components of mindfulness. So in this way, you can disentangle true effect and the expectation effect. Actually, we used another control condition in the study, in the multi-site study I presented, but one idea is to employ a sham meditation condition to better, have an idea of the efficacy of mindfulness.
So what you're saying is that there are only the five types of mindfulness meditation that you presented.
So here we had 4 different exercises, and the control condition was listening to a story. So basically, they were listening, all participants, an audio track that could be either one of the 4 different mindfulness tracks or a story that was recorded by the same mindfulness instructor.
Thank you.
We can go to this side.
Hi, my name is Gal Keshet. I have a question for Revathi. So at the beginning of your lecture, you mentioned that the triple negative cancer tends to also be more aggressive. So I understand that they're more challenging because we can't use the targeted therapy, but do you know why they also tend to be intrinsically more aggressive?
Hi, so yes, to some extent. So each of the type of breast cancers do have their own ways and mechanisms of surviving extreme conditions, and triple negative breast cancer tends to usually be a bit more metastatic, and they tend to have some survival techniques to be able to survive extreme conditions. Like in this case, we actually saw a model of cells in different stages of breast cancer, and we see how they are able to adapt to hypoxia, and perhaps this might be some of the techniques that triple negative breast cancer cells might be using to be able to survive hypoxia and other extreme conditions.
But why? Why is it connected to the triple negative?
So actually, with respect to hypoxia itself, they have upregulation of the HIF-1 alpha protein, which is the transcription factor that kind of leads all the different mechanisms that typically allows the cells to respond to hypoxia. And because of that, they are able to survive hypoxia better. Does that answer your question?
Still not quite. I understand the mechanism which enables them to cope, but not why it's connected to the fact that they are triple negative. Why it always occurs together?
I'm not able to give you a concrete answer to that, but what I understand is that the hormones, typically estrogen and progesterone, these are also known to regulate some of the different mechanisms that actually allow for survival of hypoxia. And since they can be targeted to be able to respond to kind of prevent them from surviving in hypoxia, you can treat like ER positive, PR positive breast cancers better. Yeah.
You can continue the discussion later if you still have questions. We can move to this side.
Hi. So I'm Atal from India. And my comment regarding your work was essentially that the demographics of a place really decides how much physiological demand the body has, right? So I come from India, where like 70 to 90% Indians are supposedly deficient in vitamin D, and I'm walking with intact bones. So that's why I wanted to ask. in your subjects of in your study, how do you decide what is the dosage for different subjects, given that they hail from different demographics, different continents altogether?
Very good question. So my study was a systematic review and meta-analysis. So I didn't do any interventions, but the studies that I included in my meta-analysis did. And in terms of like choosing demographics, we did try to keep it geographically diverse because we know that different populations are exposed to different variables and have access to different foods and supplementation methods. So that is a good question. I don't know what they did, but I tried to be as inclusive as I could because I did include studies, several studies from India.
All right. Thank you so much.
You're welcome. We can go to this side.
Hi, the fish guy here. I have a question to Krishnil. I was amazed to learn that like people are actually using these like GAR4 US system to actually combat cancer, because this is exactly what I use in fish or like previously in during PhD flies to like specifically target different cells and like drive like calcium indicators and these kind of things. And one of the things people do a lot in flies these days is what's called split GAR4 system. And like what they do is basically you split the GAR4 protein into like two halves and put them under the two different sort of like enhancer segment, such that basically you get sort of combinatorial you only get the sort of like a logical and and you get like high, and then like you can sort of basically target like single cell type in the brain, and like you can maybe like kill it or like monitor it. And I was just wondering, because like you are facing the same kind of problem of specificity, is this like split GAR4 kind of approach is something people have tried, or like
Very similar. I didn't want to go into like the technicalities today, but I guess the idea is the sniper recognizes antigen, and you could view that as antigen A. And if you induce a CAR or a Viper that's also antigen dependent, now it's a two antigen system that's required for the killing response to occur. And so that describes an AND gate, exactly what you're saying, but just achieving it from a slightly different way. And so the idea is the sniper might detect something either unique to the tumor, so tumor-specific antigens, or a highly expressed antigen in the brain if we're trying to localize it to the brain. And other groups have worked on things like this. And what you've said is also definitely feasible. There's also ways to extend gene circuits to become, I guess with increased complexity, people have made synthetic transcription factors you might have heard of. And so these, if put under similar systems, could be put in parallel to drive different transgenes with an anti-tumor payload. So definitely we should talk afterwards. Yeah.
That's really cool. Thanks.
Thank you.
Afternoon, I'm Leanne Kek from South Africa. My question is posed to Dr. Alessandro. I wanted to ask, how was your subject group selected, and were surveys done before the exercises? The reason why I asked this is because I wanted to know, did you detect or find out that anyone suffered from or had ADHD? Because it would have been interesting to know how the results were after the mindfulness exercises.
Okay, so the sites that participated in the data collection, the 37 sites, basically they recruited the participants, but we had some exclusion criteria. So we excluded participants with an history of mental illnesses, as the study was conducted fully online. So it's not like when you have participants in the lab and you can control the situation. So mindfulness has not really negative effects, but there are some situations in which participants do mindfulness and might feel not really good, especially if they have a previous condition of maybe depression or chronic anxiety. So we decided to be cautious and exclude those participants and not enrolling them in the experiment.
Thank you so much.
Okay.
Hi, my name is Alon. Very nice talk to all of you. Mr. Passat, I have a question for you. You presented the sniper construct as an option to avoid off-tumor cytotoxicity. And I just wanted to know if you're doing that, it will activate the cells on healthy tissue as well, or you have to still deal with the scarcity of neoantigens regardless. So how do you approach this problem?
That's very true. And it's true that with the antigens we're trying to target with the sniper screen, most of them will be tumor associated, so highly expressed but not restricted to. But we do think that when we look through, say, the Human Protein Atlas and things like this, or databases where we have antigen levels in other tissues, it's the two antigens that have to be highly expressed or there to see the killing response. So it's true that a sniper might be activated in some healthy tissue at a low level, but without the antigen that the CAR detects, the killing shouldn't occur there, so the toxicity shouldn't follow. So that's how we're viewing it, and trying to make the correct antigen pairings for that.
Thank you.
Go ahead.
Hi, I'm Rebecca, a biotech student in INSA Toulouse. My question is also for Dr. Alessandro. I'm wondering if you think that in studying mindfulness, if its recommendations could go beyond just healthy mental practices and have like applications in the clinical field for the management or even curing of more severe mental disorders.
Yes, so actually there are some protocols of mindfulness-based stress reduction, mindfulness-based cognitive therapy that are used for people that have subclinical mood disorders. So it's something that works, but I would recommend not an online mindfulness intervention, but an intervention of mindfulness with a certified teacher that can assist you and can help you. But definitely it's used also in psychotherapy, cognitive behavioral therapy as well. So definitely there are applications for more clinical populations.
Okay, thank you very much.
Welcome.
I have just a few questions. So for you, Alessandro, is there any mindfulness technique that you think is better than the others?
So from the exercises we tested, it seems that the body scan was the one that had the strongest effect. So it might be easier for participants to redirect the attention to different parts of body. So that this one seems the one that was the strongest.
And then for you, I also have a question. In terms of glioblastoma multiforme, how did you get your virus across the blood-brain barrier?
We get asked this quite a lot. Part of the answer is quite sad, to be honest. It's because with how the tumor grows and when it's detected, often patients have a disrupted blood-brain barrier. And so we've seen with more clinical work in the last decade, when tissue samples are taken, they see T cells in there. And again, it's because of the disrupted. From a more clinical perspective, some of the immunotherapy like CAR T cell clinical trials that are going on, and for glioblastoma, I think it's one of the leading ways for like exploring solid tumors now. They've gone from intraventricular or intravenous delivery to intracerebroventricular, so directly into a canal through. And so that's another way of getting a higher dose into the brain, and that's with more refinement has been shown to be safe for patients.
And then I have also one for you. In terms of hypoxia for proteins, did you find that that was better than, like, in terms of having an effect on proteins? Did it have a bigger effect on proteins or lipids?
Could you repeat the question? Sorry.
Did hypoxia affect proteins more than lipids?
I would say the most drastic change that we observed was really in the lipids. So in the IR results, we can't actually look at the protein content changes because of the fact that we need to average the whole spectra with respect to the amide-1 bond, which is the most significant bond among all the most significant peak among all the other different biomolecules. So, but we can look at the structural changes, which is what I presented. But one thing that came across consistent across both IR and morphological changes that we studied using face imaging was the lipid upregulation, which is a likely mechanism that cells usually use to respond to hypoxia, particularly because it is one of the mechanisms that these cells use to store energy, especially because they can't readily get this energy through aerobic respiration when there's a lack of oxygen. So I would say the lipid would be the most important biomarker.
Good to know. Oh, go ahead.
So I have a question for you, Crystal. So in your study for pregnant women and the risk of early labor, how long before you would recommend, or maybe during your study, before the labor, women should start to think to take these supplements?
Ideally, if a woman has any deficiencies, then right away. But in order to know you have deficiencies, you have to get them diagnosed. So it's best to go before pregnancy, which kind of is a little unrealistic because a lot of people don't plan a pregnancy. But if you are planning a pregnancy, I recommend you go now.
Thank you.
You're welcome. Any more questions?
I have a question for you, Crystal. So I was wondering that, okay, so since we know that many of these supplements have such positive impacts and we can essentially like help improve the whole pregnancy process so much better, would it be okay to just, have this set of supplements as soon as they want to be pregnant? Or is there a chance that taking these supplements too much could potentially cause some negative impacts?
Yes, that is a cautious thing that I should point out. So too much of like one of the supplements that I did find to be significant at reducing the risk of preterm birth, like selenium, could lead to toxicity. So if you need it, then you take it. So you need to be diagnosed with a deficiency. But if you know you need it and you don't have access to foods with selenium, then yes, supplementation is recommended. That's why it supplements the food that you can't access.
Thank you.
You're welcome. Okay.
If there's no other questions, I'll follow up from the great question that was asked from the audience for you. It reminded me what you just said of like, if you needed to check for nutritional levels before pregnancy, you can't really plan it. In a way, in our cancer field, that's occurrence and screening prior. Do you see any kind of platform or program that could be created to assist this?
Yes, I'm actually, thank you. I'm creating something that is going to do prediction, therefore prevention of preterm births worldwide. We spoke a lot about nutrition being very important in our last session and also about preventative care. So that is kind of like the shift that we want to go towards. It's more preventative instead of reactive. So when it's too late, you diagnose somebody, you give them meds. Whereas when it's not too late, you can do some preventative techniques. Yeah. Okay. That is it. If anyone has any more questions, we'll be around until Friday. Just find us and ask us. Thank you.